Hi, everyone. Welcome to the Rare and Orphan Disease panel. First, I'd like to thank Sachs and then my co-chair, Lubor, who will introduce himself in a second, and our esteemed panelists, who will also introduce themselves. My name is Ginger Johnson. I'm the CEO of Cello Health BioConsulting, and we're a strategic advisory firm that's focused on early innovation that largely is in the hands and the care of emerging biotech. And I want to just take 1 minute, so you can time me, to run through 4 relevant statistics that I came across in preparing for this panel that I think the themes will run through the discussion. So first, the number of rare orphan neurological disease clinical trials is steadily rising. There were 6 in 2000 and nearly 200 in 2019 and 200 again in 2020. Second statistic is that ALS is the most researched disease with a 275% increase in clinical studies from 2014 to 2020. We can talk about why. And that's followed by intractable childhood diseases. Fun fact number 3 is that we've proven we can crack this nut. There are products that have reached patients that are ranging from small molecule symptomatic therapies like Austedo for Huntington's chorea to Zolgensma, which is gene therapy for SMA, so quite a range. However, this is number 4, there's still plenty of risk. With the leading cause of failure or trial termination being lack of efficacy. Lots to unpack there. And one thing I want to submit there that likely contributes to this is that these diseases, even though small patient populations, are likely not one disease as we define them today. So I'll leave it there and turn it to Lubor to introduce yourself.
Thank you very much for this great introduction, Ginger. This is really great news, and I think it's definitely one of the topics we're going to discuss in a minute. By introduction, my name is Lubor Gaal. I'm the SVP at Locust. Locust is a global life science transaction firm. We advise biotech and pharma company on strategic transactions, also on capital increases like crossover rounds, and also strategic advice like portfolio prioritization. It's no accident that I'm on the CNS panel. I've been in CNS since the late 80s. I've been a PhD in neuroscience, and I spent many, many years in pharma BD position at Bayer and Bristol Myers Squibb, running business development in neuroscience. So I'm very pleased to be at this panel, a chair of this panel, and I forward to introduce the panelists. And maybe I start with Charles to introduce himself.
Great, thanks, Lubor. So thanks very much for having me today. So my name is Charles Stacy. I'm the president and CEO of Saracin. So my background personally is I've been at Saracin for 5 years now. Prior to this, I was with our lead investor and shareholder, a venture capital fund called Inventages. I spent 5 years there focused on the CNS space. Prior to that, some time in the US with a VC there. Originally, I am a neuroscientist by training and specialized as a surgeon within reconstructive and neurosurgery. A little bit about Saracin. So Saracin is a CNS-focused biotech company headquartered in the US and Singapore. We were founded about 20 years ago. Our lead indication up until 2 years ago was Alzheimer's disease. And more recently, we have expanded our lead compound, which is tricaprolin, a small molecule. We expanded that from Alzheimer's disease into migraine prevention, and more recently, infantile spasms, a pediatric rare form of epilepsy. So as a company, we're relatively new to the rare disease space. We'll discuss in this panel the reasons we did that, and it was for clear strategic reasons. Although as a team, we have a wealth of experience within developing diseases in this space.
Great, excellent. Thank you very much, Charles. Hans, you want to go next?
Sure, thanks, Lubor. Hello, everybody. I'm Hans Christinger, and I'm VP of Business Development at Ovid Therapeutics. And thanks to Ginger, Lubor, and Sachs for being a part of this. It's great to be here. I've been at Ovid for about 4 years now, and we're a small public company. We're focused on rare neurological disorders. The company was started in 2014 by Jeremy Levin and Matt Turing. And the idea was that, neurology, and particularly rare neurology, is at a place now where the tools, the science, the understanding, this is a field that can be cracked open now if you put the right team behind it. And so we started out, you have to start somewhere. We've got 2 core pillars that we've been working on for the last 7 years with rare neurodevelopmental disorders and then also rare seizures. And we can go into that a little bit more, but the idea is, pull together the tools, the resources, build a team that's built for purpose, translational medicine, clinical development in this area, and start knocking through the areas and build on from there. So over the last few years, we've moved a few things to the clinic. We had a program that unfortunately didn't pan out. It was one of those trials Ginger talked about in not showing efficacy in phase 3 for Angelman, but we were the first company in this area. So the field has been transformed since we started. Now you've got a whole host of people in the area. You've got new endpoints and, radically changing what's happening in that field. And we're still in it. We're working on some genetic-based modulation therapies for that area. We also moved a couple of a compound forward with Takeda in Dravet and LGS, and those phase 3 studies are starting now. And, we actually did a transaction with Takeda recently that gave us a fair amount of capital to help us accelerate our vision going forward. So we've got a series of early stage programs and that we're moving those forward aggressively and really working on Jeremy Levin's string of pearls in rare neuro as the focus for the next few years, next few months and few years. But that's a quick overview of the company. And thank you, Lubor.
Thanks very much, Hans. Jesús, would you like to go next?
Yes, hello, good evening, everyone. And thanks, Ginger, Lubor, and Sachs for this invitation. Great to be here tonight. I am the CEO of GeNeuro. GeNeuro is a company that is leveraging the biology of human endogenous retroviruses, which is that 8% of the human DNA, which is made of the rest of viruses that contaminate our ancestors. We focus on autoimmune and neurodegenerative diseases, and our lead asset, temelimab, is an antibody that is tested against disability progression in multiple sclerosis. And we already have patient on over 200 data on over 200 patients where we hit all the main markers related to disability progression. And we're now finishing phase 2 trials at the Karolinska in Sweden. The readout should be in the first quarter of next year. We also have a program in ALS, Lou Gehrig's disease, in partnership with the NIH that has been advancing well and should ready to start clinical trials sometime by the middle of next year. And last but not least, we have recently been really intrigued by the data that has been published by several groups on the link between SARS-CoV-2 and the human endogenous retroviruses and the W protein in particular, which was one of our targets. And we're working on trying to understand what this means for the post-COVID and all those patients that are experiencing neurological symptoms and some sort of neurodegeneration. So we're now also active in this space, trying to tackle this major public health problem. So we are the company focusing on human endogenous retroviruses, and this is a very large field of applications and a lot of hanging fruit from this biology that we're trying to capture.
Excellent. Thanks very much. Kasper, could you introduce yourself? You're still on mute, Kasper.
Yeah. Okay. See, that's the danger with muting. Thanks, Lubor. So I'm Kasper Roet, the CEO and co-founder of QurAlis. And as was actually introduced in the beginning, ALS is a space where there's been a lot of progress, and QurAlis sits exactly in that space. So we started QurAlis to develop breakthrough precision medicine for ALS that we can then translate into other neurological diseases. I think, if you look at why there has actually been so much progress in this neuromuscular space, it is because we're beginning to understand this disease. And it started with genetics, familial forms of the disease, providing us disease targets. Then one of the key elements that we needed is a translational model. One of our founders, Kevin Egan, a professor at Harvard, developed this stem cell technology, which basically allowed us to test molecules, bring them to patients without trying to squeeze these through an animal efficacy model that's not relevant. And the third one is access technology to get to your targets, and the ASO technology has really proven to be valuable there. So QurAlis has harnessed all these different things, and at this moment we're actually in a very exciting phase where we're breaking out of the genetic forms, the familial forms of ALS, and QurAlis really has now a good understanding of how to subdivide the sporadic forms. And Lubor mentioned that it's not even one disease; those are also multiple diseases. And once you start to solve the puzzle of sporadic disease, which is the large proportion, for instance, in ALS, that is when you can really start to help many, many different people. And that is what QurAlis is doing. We're targeting both genetic forms and sporadic forms of the disease. I'm very excited about this panel. This is my passion to talk about these things. And I'm excited about this space. Our lead asset, QRA244, targeting KV7 channel, identified through stem cell technology. We hope to have in the clinic at the end of this year, maybe beginning 2022. Thank you very much. I'm excited to be here.
Excellent. Thank you very much, Kasper. Marc, would you go like to next?
Sure. Thank you, Lubor. Thank you, Ginger, for organizing this. I'm Marc Martinez. I'm the founder and CEO of Minoryx, a company that was actually created to focus precisely on this space and the orphan CNS space. Today, we are leading the space in ALD, adrenal leukodystrophy, where we just completed a phase 3 with our lead candidate, leriglitazone, showing very interesting results, actually, and now preparing for discussions with FDA and EMA about an approval path based on the data from this phase 3 study. The molecule exploits the PPAR gamma agonism and as a mechanism of action. It's an approach that showed great potential in multiple CNS conditions, so it's a pipeline in a kind of a story, what we have in our hands. And actually, we already moved into a second indication, which is Friedreich's ataxia, where we completed a phase 2 study showing clinical benefit and now preparing for launching a phase 3. But we are also preparing for other indications as well with the same molecule. Happy to be here in such an interesting
Excellent. Excellent. Thank you very much, Marc. Last but not least, Thomas, please introduce yourself and your company.
Yeah. So my name is Thomas von Pedersen. Thank you for inviting me for this interesting meeting today. So I'm CEO of a company called NMD Pharma, and what we are focusing on are actually neuromuscular diseases, and what specifically we're focusing on is small molecule inhibitors of an ion channel called CLC1, chloride ion channel. This is an ion channel that you will not find in any other tissue than skeletal muscle, so it's highly specific. And what the small molecules will do is that they will strengthen neuromuscular transmission and acutely improve muscle functionality with a potentially broad applicability for myasthenia and SMA as our primary indications at the moment. But it could be broadened out as well as later stage, perhaps to ALS patients. We are at a clinical stage. We just completed our phase 1 SAT. So that is where we are with our front runner molecule. We have several molecules in the pipeline moving forward. So excited to be here. Thanks a lot.
Super. Thank you very much for all this introduction. And as we said, our panelists span a wide areas of diseases in this very broad topic. And this brings us to the first questions. And actually, we already heard it a little bit in the speakers. Your companies, and I think Hans said this, yeah, Kasper said it, even Marc said it, you were, and Thomas as well, you specifically were founded to work in this disease. So you're not here by accident, So what attracted you to this disease? I mean, neuroscience is not known for easy drug development. So how does it apply to rare different? Did you go into rare because it's easier, or because, to maybe Kasper's point of view, you had a unique insight into the biology, the understanding of the disease? What was the starting thesis for your company, and what got you excited about working in this space? Maybe I can start off with Kasper, because why did you get started with this company in this space?
Yeah, I started to get into this space because I wanted to help patients, and you know, there's been a lot of failures in the neurodegenerative space as a whole, and so it was really about, okay, how can we actually have a good path to success? And so when in 2011 the largest familial form of ALS was discovered, C9orf72, and parallel to that discovery, Kevin Egan pioneered this stem cell technology as a translational model into the disease. And so suddenly we could identify patients and we could test therapies in patients' own cells. So we could take skin cells or blood cells, turn them into stem cells, turn those into the neurodegenerative models, and test the molecule and see if it actually corrects the disease phenotypes. You could do that with while the patient's still alive. And so that, for me, was an enabling moment. It drove me to work with these people and with the purpose of starting a precision medicine company. Also, again, on this realization that this is actually not one disease, that these C9orf patients are very different from other ALS patients. Again, from our own group, but also from others, we see clear transcription profile differences between sporadic patients and C9 patients. And so you have to treat these patients in a different way. But how do you know if it works without a good model? And so for ALS, these things started to come together, and then a regulatory pathway that also supports a clear clinical path, a development path with clear efficacy readouts. And even biomarker readouts that you can use to measure efficacy. That all started to come into play in the beginning of this, well, the previous decade. And then in the middle of the decade, we had this first success in spinal muscular atrophy, as Ginger mentioned before. And so it really showed that we can interfere in these diseases. And so slowly, for the past decade, all the important components to get to success have fallen into place that led us to create QurAlis 4 years ago to turn ALS into a chronic disease, following the Vertex model for cystic fibrosis, one patient group at a time, and then hopefully by combinatorial therapies, being able to treat all of the ALS patients. And so that was really what drove us to start on this path. And I'm excited to see that these things are playing out actually according to our expectations.
No, you can sense that. We can sense that you're excited about this. I mean, Thomas, was that similar in your case? Did things come together? I know that you were working on neuromuscular, in your, well, you were at university or something, or so, like maybe
yes,
a little bit more.
So I actually part of what laid the scientific foundation for all of what we're discovering here and what we're working on is actually part of some academic work that was done here at Aarhus University, and we basically had some basic questions that we were addressing around this CLC1 chloride ion channel, some basic function around this channel. And we realized in that process that this was actually a target that could be used for neuromuscular disease. So we came a little bit from a different angle and then realized that this was a brand new way of improving muscle function when you have compromised neuromuscular transmission. And that has a potential pretty broad use, actually. And we then, with this new mechanism in hand, we sort of looked which indications where it would be particularly relevant, where there's an unmet need and where the mechanism of action is fitting the biology. And therefore, we went with myasthenia gravis as our first indication, which is a pure, very clean sort of disease for neuromuscular transmission defect. But we know that this is actually something that's happening in quite a large number of patient populations. And so I would say we came into this a little bit because we discovered a new mechanism, and then we realized that there are a range of indications where we could be going for, but we chose one where we understood the pathobiology quite well, and we sort of really chose a path that we think gives us the best opportunity to show proof of concept and proof of this new mechanism, while of course focusing on that this area it's a disease where there's definitely still a need for something. So those things combined a little bit, an observation, new discovery, and then sort of picking from a range of range of indications, and really sort of picking the one where the biology meets the new mechanism of action.
Yeah, well, that's exciting. That's really good. I mean, I know science should lead the way when you of course identify or select the indication for your molecule. I mean, Hans, you said Ovid was founded in order to help patients with Angelman syndrome. I mean, was there such a fundamental understanding mechanism, or was it more coming from a clinical side, understanding the patient population? What was the basis for the foundation of Ovid?
Yeah, I think there was a recognition about some of the biology and the disorder, a particular compound. There were some Angelman mice. So there was a compelling story, and there was a recognition that this was not just Angelman that we're talking about. But one of the things I like to highlight about Ovid as well is really the patient centricity, and this runs through everything that everybody on the panel here. But our example is Luke Rosen, who is our head of patient advocacy, and his daughter was diagnosed with KIF1A, which is one of our research programs today. And, he started a foundation, and, everything we do, when you've got somebody like that on your team, it really changes the dynamics and the conversations that you have. This runs through everything. So, in Angelman, we worked on some new endpoints. We worked closely with academics, with clinicians, with the FDA, with the families, and figuring out what matters, how do you measure it, and how you bring it in. So we did that in Angelman. We're doing that obviously in KIF1A as well. And and I take that even a step further. Our collaboration with Columbia that we signed last year with Wendy Chung. For me, this is a new way of developing medicines. You can look at the tagline and it's 10 targets, 10 programs, but Wendy and her team has identified 50 genetic disorders of the brain. She's a geneticist. They do research, treat patients, and patient foundations are formed around this team, right? So what you're doing is a lot of the same things that you do in drug development, but you're rewiring the ecosystem and how you work together. And just trying to paint a picture of what's a little bit different and what's a little bit special when you're working in this area, and particularly with the companies like we have on this panel.
Absolutely. And so are you saying, Hans, that as much as you understand new biology and you open the door into a new area, I mean, you also need to think of like the patients, and you maybe have to think with the FDA about new endpoints. I mean, do you think new approaches require new ways of looking at the disease, measuring the disease? I don't know. I mean, Marc, I mean, you pushing into interesting areas. Is that also what you see? I mean, you need to work on both ends, not just on the science, but also on clinical endpoints?
Indeed. Actually, you were talking about new ways. Usually that's the situation in the orphan space, being CNS or non-CNS. Many of us are the first time that the company is developing a drug in that particular disease or is using new endpoints. So the word new endpoint is a very common one in our space, which means that we need to work with the science, but also on the regulatory front, and to see if those endpoints will be really acceptable for regulators. This means that there's a lot of need of knowledge, which is what is really missing in the space, which means that you need to work closely with the people really leading the research, the key physicians in the space, but also work a lot with patients, which is also one of the characteristics of this space, to see what is really meaningful for them, and then go back to regulators to see the thoughts are really aligned with them. That's key to the development.
Very good. No, I mean, absolutely, that's challenging. Jesús, you're in a tough, not a tough, but you're in a very competitive space, MS, Linus. So, I mean, do you feel like that your drug is being, let's say, like positioned? I mean, is suffering from maybe old endpoints that maybe don't apply to your mechanism of action, because you're approaching it just from a whole new angle, is my understanding. You're not like the other MS drugs.
Absolutely. We're taking a completely different, I mean, not a different view. I mean, the main unmet medical need today in MS is disease progression, because today we have 16 drugs that are able to tackle the inflammation part, the acute inflammation part of MS, which is with the source of relapses. And today, with the arrival of the new generation of drug, including anti-CD20s, that is pretty much we have extremely efficacious drugs against that. But today we have nothing that changes meaningfully the way that the progression of the disease happens in patients. And this is what we're trying to tackle. For us, obviously it is different than the very well-established path of reducing the number of relapses. And the exchange rate between biomarkers and relapses is also pretty well known. We're opening a new path, but there's others with us. There's been the opicinumab from Biogen that was developed in parallel to us that unfortunately is now stopped, and MedDay also that opened some doors, which unfortunately didn't work, but we're still very hopeful. We basically take us, going back to why we're in what we're doing, and including, for example, in ALS, we go where the opportunities of biology lead us. We're the specialists of human endogenous retroviruses. This is what the company was created upon, because we see that there's two proteins which are made by patients, very specific to some diseases like multiple sclerosis in the case of W, which are able to produce neurodegeneration. And in some cases, it comes from the founder, of the company, basically the Institut Mérieux in France, an intern that made the relationship between the W protein and multiple sclerosis. Sometimes it comes from academia. In the case of ALS, our interest there was driven by the discoveries of the NINDS, which is the neuro part of the NIH, where they found out that the HERV-K protein, envelope K, could have a significant impact, a very important role in the development of sporadic ALS, showing that it was dose-dependent, toxic on motor neurons, and being able to do a phenotype of the disease on rodents with transgenic animal models. And we are the HERV specialists, and in this case, there was a very attractive opportunity to partner with NINDS and do the work together. And we're very happy and very proud that we're trying to bring a new angle to this poorly served and dramatic disease. We go where the opportunities of the biology are. We're really a company focused on human endogenous retroviruses, on these toxic protein, pathogenic proteins that are expressed, encoded by viral genes in our DNA. And now it's COVID. There's a neurodegenerative disease that is appearing post-COVID that is affecting a very large number of patients. And there's very good published data that is linking that to the WOF protein that we find in the neurodegeneration in multiple sclerosis and some other diseases. So that's a very interesting track, and we're following it. So for us, it's been, really dedicated to one biology that we understand. I believe we're the leaders in this field. We have a deep understanding of this biology, and we're following the opportunities to serve patients that this biology offers us.
Good. Charles, did you also follow the biology to switch from Alzheimer to, what was it, epilepsy, pediatric epileptic? I mean, it's not an obvious connection, is there, or am I missing something?
No, you're entirely right. Look, our strategy is a relatively unusual one. A lot of most of the companies here are dedicated to rare diseases. We came relatively late in the game with our company history. Throughout the course of my career, when I was in venture days, I saw a lot of companies in the rare disease space, as did a lot of my team. And I think what we saw was that the companies that weren't successful were the ones that entered this space for the wrong reasons. And the wrong reasons were because they didn't feel they had the resources or the funding to tackle bigger diseases, or they were somewhat opportunistic. Investigators, they had access to investigators or to patient pools or whatever else. So exactly as you said, and we're in good company here, that we were always determined if we're going to enter this space, we came in eyes wide open, we were going to be led by science and strong biology. And that's exactly what we did. So our lead compound, tricaprolin, addresses the metabolic dysfunction within neurological disease. The way it works is it induces a state of ketosis within patients. And those ketones then have various mechanisms, both in terms of supplying energy as an energy substrate to the brain, but also as also a sort of turning on and turning off relevant pathways within the central nervous system. So within, a few years ago, when we decided to expand our program outside of our core in Alzheimer's disease, which is now entering phase 3, we did a very systematic and mechanistic look at a breadth of indications. We had about 40 indications that we were going to enter. And we selected very much based on where we felt we would have, the biology was the strongest, the data was the strongest, and we were able to have maximal impact therefore for patients and have, the highest probability of success. What's been nice for us is that we've got a very nice sort of third-party validation of our mechanism in the form of the ketogenic diet. Essentially, how our drug works is by, inducing an elevation in plasmic ketone levels, which is exactly how the ketogenic diet works. And, nowadays the ketogenic diet is all the rage. 5 years ago, a lot of people wouldn't have heard of it. But of course, the way it works is through very strict carbohydrate and protein restriction. And therefore, causes the patient or the body to break down one's fat to produce ketones. And the area where there is a wealth of evidence, and of course, the ketogenic diet was originally developed at the Mayo Clinic in the early 20s for the treatment of drug-resistant epilepsy. So that was a very obvious area for us to go into. Infantile spasms is often second-line treatment, and in some cases, first-line treatment is ketogenic diet. And therefore, for us, it was an indication where we thought there's a very high probability of success, and a clear understanding by patients and physicians as to the clinical validation and utility.
My turn. Okay. So I'm straying a little bit, which will be fine. I'm going to ask another why question. It's really interesting to me that the modalities that are represented on my screen are largely small molecules, monoclonal antibody with GeNeuro, and then antisense with QurAlis. If you talk to some people, they might say the future of rare and orphan disease is gene therapy or other gene modulating technologies. And I'm sure that you've chosen your modality, as many of you said, the science leads the way. You're an expert in this disease biology or a particular pathway, so you choose the approach that fits. But still, I'm curious as to what you think about how you fit within the future competitive set. And is there room for everyone, or is there actually need for everyone in terms of complementarity for these diseases? So maybe I'll start with Kasper again.
Thanks. It's an honor to be the one to bite the. So a couple of comments to that, and you're very accurate. Well, we do both small molecules and ASOs, so antisense. There are things that you can do with antisense that you cannot do with a gene therapy, at least not yet. We set up this company, our company, to be disease focused, which means modality agnostic. And so I wouldn't say that we're never going to do gene therapy. We considered it not really ready for prime time for large cohorts of patients. There have been a lot of issues. I'm actually trained as a gene therapy specialist that work together with Unicure, with Galapagos, so adeno, AAV, to develop spinal cord injury therapies. And so I know that field quite well. I also know the limitations. And so it's very hard to regulate gene expression. And there is also a transient decrease usually in terms of levels. There's toxicity. There's only a one-time treatment, and then you get an immune response against it. Most importantly, there's huge production issues. And so if you want to make a lot of viral particles, and if you want to spread it out, you need a lot of them, then it's very hard to make a lot. And if you then have a lot of patients, it's very hard to treat them all. And so in a way also we see that the modality is just not there at that same level as where we would like it to be. And so for the future of QurAlis, I do think gene therapy will definitely be part of the future of QurAlis. But for our specific targets, for our specific disease mechanisms, they actually are not the preferred modality. And plus, we see then also a lot of problems with which are still associated with gene therapy. I'm very happy it works for some patients. and for some diseases, but it's usually very small populations. And even then, I know that there's a lot of issues just getting the right batches and having good quality control on that. And so there's challenges there. And I know that people are trying to solve that by, building very large factories in China or, all over the world, but no one has really cracked it yet. And so I think that's what I would say. I think the thing that you don't mention is CRISPR. And so and again, I think I mentioned this when we spoke once before. One of my SEV members, Chris Shaw, said that he cures families by embryonic selection. And if you're talking about the ultimate therapy, if you have a familial gene that is causing disease, but you're actually able to, in offspring of that family, select those embryos that don't have it, then you've gotten rid of the disease altogether. I think the next best thing would be CRISPR, but we're also not there yet. And so depends on your target, I guess, is my short answer.
Yeah. And I don't have to ask everybody the same question, but really good points and about the manufacturing and the business model. That's one more thing that you've got to get down. It's not just the clinical and the regulatory and the endpoints and the biomarkers and blah, 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 but you got to be able to get it to the patient. But so it's a twist on this question for Tom. You have a small molecule that is a pipeline in a molecule. What did you say? A pipeline in a drug? Whereas gene therapy you can think of as almost the opposite, right? It's one approach, one patient population. What any comments on that and the two approaches?
Well, I think so. I think the example I would sort of choose to discuss this point in is really SMA, because we have in that space we have seen 3 new ways of increasing SMN protein, which has been, it's a fantastic time for SMA patients. But what we're also realizing is that these patients still need more treatment. So then I think the likely situation is that while we can get a long way with these new techniques, I think, and new treatment approaches and gene therapy included, I think there will be a layer on top of that where we need to find additional treatment approaches to really help the patients best possible way. And there we may need to find treatment approaches that are a little bit like what we are actually doing. Some treatment approaches that can go a little bit broad in their approach, provided that we are well informed about the remaining deficit. And here we can use specific biomarkers for assessing whether there is a need in these populations, because the reality is that with SMA, now they have new treatments, we're almost seeing a completely new clinical phenotype, and we need to learn about that new clinical phenotype. And there we need to sort of have biomarkers to know what is the next layer of treatment we need to give the patient. And my strong view here on that point is that SMA patients treated with SMN upregulating therapies, they are definitely improving, but we need to do more. And there are some peripheral aspects in that are not handled yet, and that leads still to some deficit. So I think while these gene therapies are very promising, we're not the full. They're not all the way. I mean, we need more. That's how I see ours fitting in.
Okay, thank you.
So I think in general you will need different treatment modalities.
Mm-hmm. So Hans, maybe this is a good opportunity to ask you about the recent deal that you did with Takeda. It's quite interesting, but I think I've read what that gave you is some spending cash. So you're looking for opportunities. What are you thinking about modalities there? Are you just going for the right fit, or do you think about, can I take this to the market alone? Am I going to have to partner? I'm sure you think about all those things when you're looking.
Yeah, and I guess maybe I can answer a little bit of the last discussion too with a couple of examples that we have. But we've got a good balance now, and looking for bringing things in, accelerating our internal programs and bringing in complementary programs as well. We've built a fantastic translational medicine clinical development team, and can we take advantage of that and the position that we have to move things forward and really accelerate our vision. To go back to an example, and we're therapeutic modality agnostic, right? Our first two programs were small molecule just because that's the way things started in the history of the company. But most of our programs right now are RNA modulation, whether it's short hairpin RNA, microRNA, a bunch of different modalities. And if you look at gene therapy in Angelman, if you're not able to control the expression tightly enough, and if you miss, you'll actually cause duplication 15q, which you don't want to do. So we were never big on gene therapy for that particular approach. We see it in the future, just like Kasper articulated. And even with the different therapeutic modalities, the ASOs that are out there now, these patients are often 4 years old, 2 years old by the time they're diagnosed. So it's not going to be a cure. So you're going to still need some other tools, if you will. And this was the idea of our OV101 compound as well. So, it was, we believed had a lot of promise to really impact the syndrome. And even if you had the ASOs, this would be complementary and you would need it as well because you'd have residual effects. So that's one example. And if I look at KIF1A as well, there are certain mutations that are devastating where the children don't last past 2 to 3 years old. And there, the direct genetic fix in whatever fashion it is, whether it's gene therapy or shRNA or whatever, that makes a lot of sense. But you'll also have other mutations that may not be picked up by that particular therapy, and they're less severe. So the people live a little bit longer. And you can see these mutations they're perturbing microtubule interactions or something like that. So I think what you'll see is you've got the insult from the genetic disorder, and then you'll have, the brain is tightly regulated. So you're going to have stuff, whether it's the actual protein or what is moving, is in the wrong place at the wrong time. And that's going to trigger the body to react. And there you might have a whole lot of different medicines that might work. Some of the things Kasper is working on could be applicable for KIF1A, for example, in some of these patient populations. So, we really try to follow the science. The nice thing is that, in many of these cases, what the gene is that you're targeting, and you've got some models where you can show that you've got target engagement. And you can try different therapeutic modalities and parallel them before you make huge investments. You can figure out which ones you're going to move down the path on. And, you're doing this in an area where the patient is totally engaged. So you've got natural history, you've got samples, and you've got speed to testing things in the clinic because everybody's in there with you, right? So these are some of the things. It's complicated, but these are some of the benefits of working in here. And hopefully that tells a little bit of how we think about it.
No, thank you. That's very helpful. I'll ask Marc the same question and then open it up to Charles and Jesús if you want to chime in. So I'm watching the time. I want to give Lubor a chance for the last question. But Marc, I'm turning to you specifically because you've got at least Bluebird and Swan Bio right there in your space with gene therapies. I'm not sure how far out, but when you think about the future competitive environment, how do you think about that?
Let me start being a bit more provocative than my colleagues. So while we wait for the bright future that the gene therapy is going to bring, there's a present where patients need a treatment. And that's the issue that I see with the gene therapy. I see that as a very promising approach in certain diseases, not all of them, but it's going to take long before there's an approach that is broadly applied. And also even once approved, it's not that easy to apply broadly in the world. So it may be available in certain countries, but not everywhere. So I think that really other approaches are going to be needed. And I think that there's going to be room for many different approaches. And I see more combinations at the end of the day. We are talking about very difficult disorders in most of the cases. So I don't see many therapies out there really being the only therapy really curing completely patients in this space. In our specific field, ALD, you mentioned two good names, so Swan Bio and Bluebird Bio. First, it's important to differentiate. In ALD, there are two forms of the disease: the chronic form of the disease, called AMN, and the acute form of the disease called CALD. Lubor Bio has a very nice approach, ex vivo gene therapy, an alternative to the transplant, but only useful for CALD. And SwanBio is working on the chronic form of the disease. So really trying to solve the spinal cord disease. SwanBio is in preclinical stage while we finish phase 3. So I think that we have a clear opportunity. We could be first to market if we can make it to the market with the data we generated. So it's a long time. And when we compare with Lubor Bio, our molecule also has an effect on the acute form of the disease. Ours is an oral treatment taken once a day that can be is much more convenient. That so far show prevention or reduction of patients transitioning into the acute form of the disease. So if we can avoid the need of a transplant or a gene therapy, I think that would be a win for patients and for the health as well. So at the end of the day, I think that the gene therapy is going to change dramatically the space in many different indications, but it's not going to change everything. And I think that many different approaches are going to be needed. Some of other alternatives have been mentioned by my colleagues, which are also very, very attractive. And in certain diseases, even more attractive than gene therapy itself, in my opinion.
Thank you. So I'll open it up to Jesús or Charles. If you want to comment on modalities, please do so.
I completely agree with Marc. They are, we have a toolbox, and the important thing is the patient and how to bring the best and possible fastest solution to the patient. We're using antibodies in neurology, which means obviously that, when the mode of action is in the brain, we have a lot of it that is circulating and not making it to the target. But on the other hand, antibodies are extremely safe, well-known drugs, today not that expensive to produce. And when you are targeting a protein that has no physiological function, it helps you, have a very, very favorable safety profile, which makes you be very happy and very, confident for treatments that are chronic treatments for patients on the very, very long term. And you need to think about that when you're looking at how you are treating the patient and what is in the best interest of the patient. And again, there's different tools. There's also a little bit of fads in the sense that, we've all been long enough, all of us around, to know that there's also the fad effect. For a while, it's going to be red, and then it's going to be blue, and they come back to be green. And we've all been long around enough to see how the colors change. And yes, today the flavor of the month is, but I think that we should all concentrate on what is the best way to address the target. And we have a toolbox. We should use it. There's not one size fits all.
Charles?
Yeah, I mean, for me, look, not a lot to add. I'll just say, look, I think it's going to be hugely disruptive. It's going to have a massive impact, which is just great due to the unmet need. I think all of us, clearly aware of companies that need to keep an eye on the gene therapy space, but I think it's going to be very positive for this whole space. And for us, and I think it's going to be very dependent upon the indication. For us, our mechanism, leveraging sort of the data that exists within the ketogenic diet for us was, means that hopefully the probability of success is very high, and therefore, it's a de-risk pathway, which ultimately is going to provide good options to patients and to physicians. So but no, I think it's going to be a very important area for us as an indication space.
I agree. Thank you.
So I'm going to pack a few questions into my question, and I actually opened up to the entire panel to speak about it. And I will ask, there are, for instance, like the one common problem that you always hear when you have a neuroscience panel, is that animal models don't translate into human disease. I mean, that's basically, you can hear that from any panel. But I was very happy to hear that Kasper and Thomas have models now that, are coming out of research, which they think don't have that problem. They are actually translatable into human disease. And so I would like to talk to them a little bit. And I would also then open up to all the people saying, how do you overcome this issue then? We talked about you have unique insights into the disease. How did you come about this? I mean, if animal models didn't help you, was it genomic studies, patient samples? How do you then, translate, that into your research and really apply? And the ultimate question is, then how do you translate into the early let's say, I don't want to call it clinical proof of concept, but is there any way that you can construct your studies that because you have that unique insight about the biology, about the disease, or whatever it is, that you can maybe have a readout in the clinic sooner than what maybe would be classic phase 2b, double-blind, placebo-controlled in hundreds of studies? So how does it all fit together? And maybe we want to start off with either Kasper or Thomas, because I thought it was really interesting to hear that you had some unique animal models that you think are fully translatable.
I'm happy to jump in here. So we in part actually chose the indication based on that. So the first indication, as we spoke about earlier, we have several different indications we could be pursuing, and we chose actually one. Since we're the first to pursue this particular ion channel as a drug target, we wanted to be confident that what we were seeing in the animal model could actually be translated to patients. So one of the arguments for actually choosing myasthenia as our first indication was actually that the animal models are known to be translatable. These animal models do really very well replicate what we see in the patients. I would say and Kasper can comment on this, but I think that the situation on animal models for ALS is much more complicated there. But for myasthenia, it's actually incredibly good, and that has definitely guided us. On top of that, we also chose these models, and we know because the biology is well understood, the disease mechanisms are well understood, and then we have translational biomarkers that we can use in animal models and also use them in the clinic and really guide the selection of indication and also use them as efficacy readout. Those are that's how we kind of bridge this and how we could use animal models. But I would say you probably have on this panel, you have a situation where maybe one of the best animal models available in rare diseases, at least in my view, the myasthenia animal model is very, very good. And then the ALS animal models have been complicated, but I'll pass it over to Kasper to speak about that point and how they then were able to overcome that challenge, because ALS animal models are complicated.
Yeah, well, I thank you, Thomas. I'm not saying all animal models are bad. Like, if an animal model works and it translates to the clinic, great. And sorry for the animals, by the way. But then you have something that you can use. In ALS, the motor system, which is the system that's degenerating, is wired differently in rodents than it is in humans. So that's one of the big problems. The other one is, the only motor neuron degeneration we can create in animals is by highly overexpressing mutated proteins. And that doesn't seem to translate very well. Maybe the SOD1 model is the exception to that. But what we found is we had, in these stem cells, we had a dysregulation of potassium channel Kb7. It led to an excitability abnormality in the stem cell models from patients. We didn't find that in the animals. So then what do you do, right? And so our study from our founders, I wasn't even part of the consortium at that time, was really to try and take a molecule directly to patients then. And it was lucky that the ALS Association wanted to support that financially, that the clinical organization in the United States, NIELS, was also kind of tired of failures of the animal model, and that GSK at that time wanted to break boundaries and do something new. And so they actually all came together in the Harvard Stem Cell Institute and really brought a molecule on that target to the clinic without animal data. And that study results from that clinical phase 2A biomarker study was published in December last year. So obviously it took a couple of years to really get to the answer, but it showed that the predictions from the stem cell models on biomarker effects, but also at least on the biomarkers tied to disease progression, like a CMAP, compound muscle action potential measurement, showed that actually correlated to exactly what we found in the stem cell model. So for us, that was a validation that the stem cell model can be translated to the clinic, and you don't need the animal model. And now, after that, during this time, actually, we started to understand the disease so much better. And now we also know that there's a lot of genetic regulatory differences, non-coding RNA differences between the animals and the stem cell models. And honestly, the field has wasted a lot of time trying to reconcile what was found in animals with what was found in the stem cell models. And now we kind of have accepted that you cannot reconcile it, that there's a good correlation between the stem cell models and the patients, and that the animal models are mostly different. And once you've found that out, then you know that you found your new path basically, and that is the path that QurAlis has taken.
So maybe just a follow on there. So we're using the myasthenia model to, of course, to pursue that particular indication, but we're also using it to validate some biomarker studies, some biomarker signals that subsequently can be explored in patients. And once we've qualified the treatment approach for that particular biomarker signal, that can then go kind of broader at the patient level and see whether they have the same biomarker signal, and then go, rather than go via the animals, actually go and do clinical observational studies, use that biomarker study to know which population studies, which patient populations we could actually be addressing afterwards. We're using one animal model to really generate the proof of concept for the treatment, but also for the biomarker that we can then use in the clinic to different indications. So that's kind of using the best model to get a proof of concept ready and then broaden that out in the clinic. So that's one approach to overcome the bad animal model.
Yeah. And the other panelists, I mean, are you using also biomarker approaches to bridge that gap, or are you using genetic markers to identify patient population, or what is that? How can you get faster confidence that you're actually treating the right patient or your drug is actually working? I mean, what are your strategies?
For us, it's both.
Yeah, it's both. And, I think Kasper articulated much better what I was going to say. So I think we look at it the same way. It's where you've got animal models, you use them to the best ability you can. Where you don't go and create an animal model. You show that it works in iPSCs or whatever format that you've got target engagement. You show it's safe and you go to the clinic. We've always tried to have biomarkers. Our soticlestat program, we had a nice PET ligand that Takeda put together. So for us, it's always important to try to make sure you've got a total package like that and are moving along following the science.
Yeah, us too. I mean, we use that animal models before we went into humans, and I think we recognize the limitations of them. But ask me in a year's time whether I translate into the clinic, because we're still in our first clinical study. But recognizing the limitations, they were material for helping our internal decision processes and deciding our path forward and selecting that indication. I think I agree with Hans in that point.
Excellent. We have 2 more minutes or 3 more minutes, and if there's anybody in the audience who wants to ask a question, please do it now. I mean, Ginger, do you want to maybe lead off with the last question?
Yeah, there was a question, but I don't know how to find it.
I think it was about ASOs, the future of ASOs.
Oh.
No, I mean, but one thing I would like to know, I mean, we already talked about you came in here because you had breakthroughs, understanding in neuroscience. I mean, one thing that, oncology has, so there are 2 big trends, of course, that are going on in oncology. And I heard a little bit of this already in our panel as well, but I want to see if you see any kind of such a seminal event happening anytime soon. One is the immuno-oncology area, so how important is the immune system in neuroscience, and the other one is precision medicine. And I heard somebody saying we're doing precision or neurology. Do you think it's going to be both in neuroscience, one or the other? If you had, if you had the crystal ball, or why don't you look in your crystal ball, and what do you see going forward happening in rare and orphan neuroscience? Anybody? Nobody?
Yeah, there's absolutely no question. I mean, this is, walking through an open door. I think that there's absolutely no question that our better understanding of the immune system in the brain is going to help us in breakthroughs in many diseases. Not only for us in multiple sclerosis, it's clear that the neurodegeneration is driven by a slow cooking immune process within the brain long term. Now we're looking at things, there's a lot of interesting publications in the relationship with the immune system in Alzheimer's and others. So yes, it's definitely a trend today. How will that be leveraged into the treatments? We know the ones we have, but certainly there is a lot of new opportunities in that area coming up.
Excellent. 1 more minute.
My future prediction is precision combinatorial therapies for neuroscience, exactly the same as it is in oncology.
And the payers need to get ready for that.
Well, I will leave that question for the future.
Now that we have 30 seconds to tackle that one.
Very good. So maybe that's a good way to end the session. So I really want to thank all your panelists for a really interesting discussion. I could have, of course, continued for another hour easily or so. I found it very fascinating. Also, I found it really exciting to hear that new insights are having been generated and we leverage them for the benefit of patients. So I think it's important that we find new ways to crack the tough nut the brain is. So thanks very much for all your discussion and participation, and I really enjoyed having this discussion with all of you.
Me too. What he said.
Thank you for inviting us.
Thank you.
Thank you very
Thanks,
much.